Welcome everyone. Thank you for joining us today in the second special lecture of this year in the Progress and Prospects in Biology special webinar series. And thank you all for continuously supporting us over these few months. I am Nandita and I will be your moderator for today's session by Professor Rajib from Jadavpur University. So before we begin, I would like to remind you that you can post your question in the YouTube chat box and we shall moderate them at the end of the lecture. And the feedback link will be provided in the chat box near the end of the session and will not be shared via email. So now I would like to introduce our speaker. So Professor Bandyopadhyay is the professor in the Department of Instrumentation Electronics Engineering, Jadavpur University, Kolkata, India. He did his PhD at Jadavpur University in the area of intelligent control. Professor Bandyopadhyay joined the Jadavpur University in the year 1990 and is involved in teaching and research since then. He has guided 18 PhD students and 8 PhD students are working under his guidance at present. His research areas involve NIR and Raman spectroscopy, machine olfaction, electronic tongue, and electronic nose. His research group has developed electronic nose and tongue system for quality evaluation of tea. Professor Bandyopadhyay has published more than 90 research papers and 5 book chapters in reputed international journals. He has been invited to deliver talks in many national and international universities across the globe. He has presented more than 100 papers in national and international conferences. Professor Bandyopadhyay is the member of many national committees of CSIR, UGC, universities and engineering colleges in India. He was the president of the Kolkata section of International Society for Automation, ISA, during 2015 to 2016, and the faculty advisor for the students chapter of ISA at Jadavpur University. The research group of Professor Bandyopadhyay has collaborations for Center for Development of Advanced Computing, CDAC, Kolkata, Toklai Tea Research Institute, National Environmental Engineering Research Institute, Nagpur, and ITMO University, St. Petersburg, Russia. He is a research professor at ITMO University, Russia since 2015. So a very warm welcome, Professor Bandyopadhyay. We are glad to host you today. So now I would like to hand over to you for delivering today's talk. So over to you, sir.
Thank you, Nandita, for this introduction, and very good evening, afternoon, morning. I don't know what are the other things in terms I should add, because in this pandemic time, it's altogether a new experience for us while addressing. And warm welcome to all of you who are present. And you see, I'm from the introduction, you have understood that I'm a person who is far from biology because I did my engineering in electronics field, but now venturing in the area of sensors, and which are these sensors are they are now being used in the agriculture industry, particularly these botanists, and they are using this type of sensors and sensing systems. And we have close collaboration with this agriculture and departments and labs, and the users. And now we are trying also to explore the sensors, because the sensing principles are almost same. So for biological applications, we can develop the sensors, and this can be used very useful for that. So I would like to thank Professor Banerjee, NRI Banerjee, for inviting me in this in this series, webinar series. And it's a long series. She's organizing quite successfully with her students, and I'm very excited to present our research in front of the audience like you, because most of you are from biology background. So if you can find some ideas where we can collaborate, we shall be very glad. Okay, so with this introduction, let me start my presentation. And yeah, Is the screen visible? Okay. Uh,
Sir, it's visible, sir.
Okay, okay, thank you. So I've chosen this topic, that chemical and allied sensing system for application in agriculture, and we have been working in this area for the last 15, 16 years or so, and our research work started with the input from the tea industry. In the tea industry, the tea is tasted by the tea tasters, and they evaluate the quality of tea, and based on that, the price is fixed. So there is a requirement, and the method is purely subjective, and the tea industry, they are in the lookout for a system which can I mean, objectively estimate the quality, which is really not very easy. But anyway, there are some ways to, I mean, go in that direction. And we started our work with the development of electronic nose. But before that, let me just, since my area of application is agriculture, let me just go through about about Indian agriculture, where we have been targeting for to develop the sensors, and what are the problems, and what are the uh, issues. So you see, the main problem, or the main reason why we have chosen agriculture is that it provides the employment to around 56.54% of Indian population. So you see, there's a huge, population. They are, and the population of India is so, large, that and a lot of people, they are based on this agriculture. And whenever our agricultural production is good, then our economy is good in that year. And if it is bad, then it definitely affects our economy significantly. So agriculture is the mainstay of Indian economy, and these are some of the problems which I have highlighted. And this fragmented ownership, this is one issue, and stiff competition among entrepreneurs, and very little protection from forces of nature, and that is one major issue. If there is a, good rainfall, then we get good production. Very little automation and standardization, because we have huge, unskilled or skilled laborer. So they do not feel the urge to employ these techniques, but these are necessary for maintaining quality. Lack of right information, for that these computer systems or these mobile systems may be useful. There are some systems for that, but they're not really there's a lot of requirement, more requirement is there. Few or no sensing systems in the hand of farmers. There are only a few sensing systems for soil card and all that, for measuring the soil parameters, and the other, whether a particular plant is affected by some pests or something like that. There's almost no mechanization and automation. This I've already mentioned. So, and the national status, see, this ICAR, this Indian Council for Agricultural Research, they have so far, after independence, they have contributed significantly in terms of producing different types of seeds, and different type of varieties of the crops. And that's why there is no famine after the independence in India. And that credit entirely goes to the agricultural scientists. But there are some limitations, and particularly the high-end technologies that are yet to be employed for this purpose. Rather than there has been rampant use of this agriculture, these pesticides and these fertilizers, which has really reduced the fertility of the soil. So there are some issues on that. Although Indian government, they have taken very important steps for taking this into account. And this is the international status. High level of mechanization is available, and the productivity is also is more with less manpower. And the problem is that they are particularly suited for those advanced countries. They're expensive, and the technologies are also guarded. So we have to develop the technologies of our own. That is the need, and that's why India government, they have published one document. It's called Agri Vision to 2050. So for the next 30 years, what would be the roadmap in the field of agriculture? So it's a big document, and I've taken some portion of this that to the point that I have highlighted, and that it is needed to see the leverage the advances in other sciences, engineering and social science, to enhance agriculture research. And in this domain, we have been trying for to develop some products which may be useful in the Indian agriculture in the industry, as well as the scientists. Okay, so the sensors and ICT, these are the possibilities for the preparation of soil and sowing of seeds and plant, and irrigation, application of fertilizers, application of fertilizers and pesticides, harvesting, storage. In every stage, sensors may be employed, and which are necessary, because all most of the things are done, manually. The checking or the quality assessment, all these things are done manually. And market in every during transportation also, some sensors should be there in the boxes, so that the quality is maintained. And finally, the food safety, that's a big issue nowadays. So we need good sensors, sensing systems for that. Okay, so in this with this brief introduction to agriculture and the requirement of Indian agriculture, I'd like to highlight the the topics today, which I'm going to discuss. This is electronic nose. I'll start with electronic nose, and we have developed this electronic nose for this tea quality estimation. Then we have developed electronic tongue and the other type of sensors, and now we are also exploring the spectroscopic sensors for deployment in the field. So we start with electronic nose, and the first slide that I show everywhere in while giving this lecture on electronic nose is that, see, electronic nose, it's basically to mimic the our human nose. Okay, and human nose is one of the five sensory organs. We have this eye, ear, skin, nose, and tongue. And out of these five sensory organs, we have the scientists and engineers, we have been trying to mimic these five sensory organs for quite some time. And we have and out of these five, the first three have been mimicked very successfully, and powerful instruments and machines are available. For example, we have the camera and the microscopes, which are more powerful than our human eye. We have the microphone, which are much more sensitive than our human ear. And we have the touch sensitive devices, or the mobile phone, you see the touchpad. So these technologies are quite established, but the technology for mimicking nose and tongue, we call it electronic nose and electronic tongue. These two are really in the new entrance in this domain. And scientists have been trying to mimic the senses of smell and taste, but it is still we have to do a long research for quite some time. This field is not really very new. For the last 30-40 years, the scientists have been trying to mimic this nose and tongue. And another major issue is that most of the solutions, they are application specific. So for example, if you have an electronic nose, that will be suitable only for tea. That cannot be used for say fragrance, or the measurement of quality of wine. So we have to have another system for that. Okay, so generic electronic nose or generic electronic tongue is still far from reality. Anyway, so with this introduction, I'll tell you about this electronic nose. It is basically, some of you might be knowing what is an electronic nose by this time, because it has become quite popular, and it is now being used in the food industry, particularly in the food industry, and also in the wine industry and perfume industry. And it's basically comprises of a sensor array, and the sensor array is the heart of the electronic nose. And in the sensor array, in earlier days we had say 6 sensors or 8 sensors or 10 sensors, but nowadays the electronics nose, and they are coming with large number of sensors. And if you have a large number of sensors, then we can have a large number of applications or the applications are much more accurate, or the estimations are much more accurate. Now, one major issue for this sensors are that these sensors are broadly tuned, or they are not specific to a particular compound. For example, say the sensors are available for, say, oxygen or, say, carbon dioxide. These are very specific sensors, but the sensors that we use for electronic nose, and this electronic nose is used mostly where the complex mixture is there. Okay. So, for example, in tea, there are hundreds of compounds which are responsible for the flavor that we get out of tea. And for that, if you have to develop the electronic nose with all these for all these compounds, specific compounds, then we need, say, a few hundreds of sensors. That is really very difficult. But rather, the idea is that it is similar to the idea of our human nose, that we have the non-specific sensors. That means each sensor will respond to the different chemicals in a different way, and a pattern is generated. And this pattern, nowadays we have the very strong this machine intelligence and this artificial intelligence techniques are available. So if you have a large huge information, then that can be deciphered, and we can identify that particular compound, the quality of that particular compound. So this is, in a nutshell, the idea behind this electronic nose.
Sir, sorry to interrupt, but there is a taskbar coming regarding the stop sharing slide, and hide option is there. So if you could please click on the hide op. Okay, sir.
Okay.
Thank you, sir.
Thank you, Nandita. So this is the heart of the sensor. This is the heart of the electronic nose, and see, this is a sensor array, and the sensor array must be exposed to sensor array it must be exposed to this odor molecules. So for that, there is an odor delivery system. It is just like our human nose when we sniff the odor. So in electronic nose, we have the pump. So that pump delivers. It is responsible for delivering the odorant molecules onto the to the sensor array, and then the sensor array responds. It gives some electrical signal, and then it is conditioned, and data is, and this acquisition takes place. After that, signal is processed, and then we have this pattern recognition. So most of the modules are more or less, there is not much of research component in that, but research component is there in designing the sensor array chamber and this pattern recognition. Other blocks are more or less standard. Now, about the application, there are several applications of electronic nose. There are many research papers, starting from environmental monitoring and medical diagnostics also. And there are some applications. I have seen one paper where one scientist, he has studied the kidney patient when he is undergoing dialysis process, to just check the change of odor from the person who is undergoing dialysis, because a kidney patient has a large urea content, and because of that, the smell, there's a very bad smell coming out from his body. So that was monitored by the scientist with the help of electronic nose. So there are many applications. Wherever there is some smell or some volatiles coming out from some system, we can employ electronic nose. But remember that this electronic nose is used only where there's a mixture of compounds, and when it's particularly when the mixture is complex. But if you have only one or two gases to detect, or one or two volatiles to detect, then we can use only a particular sensor for that. That is not called an electronic nose. But one of the most important, or the wide area where electronic nose is used, is in the area of food and beverages. And this electronic nose has been used for quality monitoring of all this food and beverages, tea, coffee, cold drinks, meat, fish, and so on. And for aerospace application, NASA used one electronic nose to monitor the internal environment of the aerospace, and that traveled in space quite some time back. And another application area is the cosmetics and fragrance industry. And in this way, France is a pioneer, and there are a number of manufacturers in France for electronic nose, and they're regularly using this electronic nose for fragrance industry and this wine industry. Detection of explosives is not really very successful so far on electronic nose, although there are some researches. Now about the sensors, there are different type of sensors for this electronic nose. This is the first one is the MOS sensor, or this metal oxide semiconductor sensor. And this metal oxide semiconductor sensor, when this is exposed to the volatile compounds, there's a change in resistance, and that that change in resistance is somewhat related to the concentration of the odorant molecule. And this is the conducting polymer. This is another type of resistive sensor. And but the most popular type of sensors which are used in electronic nose is the MOS sensor. One example of the MOS sensor is that it is used by the policeman to check the drunken drivers. So this that is quite successful, and that is quite stable. And one company in Japan, it's Figaro. They are manufacturing these MOS sensors for quite some time, and their sensors are very stable, and time-tested sensors are there. Okay, but one major advantage is that they need a large amount of power because there is a heater inside it, and that MOS sensor must be heated to a temperature of, or the surface of the sensor is heated to a temperature of 300 degrees centigrade. So that consumes a lot of power. But even then, these sensors are mostly popular, and there are other type of sensors. For example, there's a change in capacitance or change in voltage. And another sensor where we are, we can make in our laboratory is the quartz crystal microbalance sensor. It's basically there's a quartz crystal, and the frequency of the quartz crystal changes if a particular odorant molecule is adsorbed on the surface. Okay, so if the concentration is more, there will be more change in the frequency. So we can coat with a particular sensitive material on the quartz crystal. So that we are regularly doing in our lab. There are other sensors. And there's a change of temperature here. There's a change of wavelength. Surface plasma sensors are also very popular, and they're quite sensitive. Okay, so these are some of the photographs. This is the photograph for metal oxide semiconductor sensor, and see, this is the sensor surface, and there's a sieve over it so that the dust particles or the bigger particles, they cannot come in contact with the sensor surface. There's a heater inside it, and this is the quartz crystal microbalance. On this quartz crystal, we coat a particular material, and that particular material, we select it in such a way that it will absorb only the target molecule. And this is the conducting polymer. This is also used in the research labs. One major advantage is that it operates in the room temperature. We do not need much power for that. Now, this is the basic principle of this MOS sensor. The idea is that when it is heated, then the oxygen molecules, they are stuck on the surface, and they are stuck as an ion on the surface here. Okay, at high temperature. And when the reducing gas comes in contact with the surface of the semiconducting material, then it interacts with this oxygen ions, and this oxides are formed, and there is a release of electron. So this release of electron means the conductivity is more, or the resistance is less. So if the more number of volatiles are there will be more number of electrons, and the change of resistance will be more. And from there we can calculate, see, the number of electron that is coming out is somewhat related to the concentration of this reducing gas and the number of molecules of the reducing gas. So from there we can estimate their concentration. And interfacing of MOS sensors, I'm not going into details, but honestly speaking, if in our electronics lab, it is very easy for us because we need only one resistance in series with the sensor, and that this is the resistance. And if you can measure this voltage here, then we get the concentration. So this voltage is connected directly to the computer with the help of some A to D converter. So we get the data from the sensor directly. So it's really a very easy way to connect a MOS sensor, and we are regularly giving this project to our undergraduate students for making an electronic nose with say 4 or 5 sensors. They buy it from the market. Okay, so this was the first prototype that was developed, and you see there's a fan here, and this is the sample holder where we put the T samples or some other sample for odor collection. And there's a pumping arrangement here by which the odorant molecules are taken to the sensor array, and the sensor array is here, and they're dragged by the fan and to take so that the odorant molecules, they hit the surface and then they go out. And after some time, there's a mechanism to purge the sensor. So this is the screenshot of the electronic nose. This is for the T testers. So after we have designed the electronic nose in such a way that the T tester will give a score from 0 to 10. That is usually employed. This type of T tester sheets are there. So they imply they say that if it is 6, then the price will depend upon that. If it is 8, the price will be more, something like that. So we try to correlate the T tester score with the sensor response, and partially we were successful. We published some papers, but I'll tell you the story after some time. And this is the quartz crystal microbalance sensor that we are making in our lab. This is the quartz crystal on which we coat a particular material, and these are the advantages. And see, this is the basic principle of this quartz crystal, that the frequency, the change in frequency, the resonant frequency. See, this quartz crystal are used in watches and other this microprocessor or microcontroller boards. So their resonant frequency is fixed. It's when they're sealed, then this resonant frequency is constant. And but here we are not sealing the quartz crystal surface, but we are exposing this, we are coating it with a certain material, and then we are exposing it to the volatiles. And because of this absorbance of these volatile molecules on the surface, there will be a change in frequency, and that is governed by an equation. It's called Sorbury equation, and it's directly proportional. One major advantage of this quartz crystal microbalance is that, see, the name is microbalance. So it can it's meant for measuring very small amount of weight. So if there is a change of mass of 1 nanogram, that also it can detect. And because of the change of 1 nanogram, we can have, say, frequency drift of 1 hertz or so. And if it is resonating at say 10 megahertz, now the electronic circuits are available by which we can measure that change accurately. So that we are regularly using in our lab. So if some of you are interested, you can come and see the facility. These are some of the coating techniques by which we coat on the quartz crystal. We generate a high voltage. It's called electrostatic spraying, and it generates a high voltage. And because of that, there is a very fine spray on this crystal, and then the coating is done. This is with the help of nebulizer. Nebulizer also is used for coating the sensors. This is the nebulization coating. So this is the quartz crystal array. So here you see there are 8 such sensors which can be coated with different materials to make an electronic nose. And this is the prototype version of this quartz crystal microbalance. We have used this computer cabinet for making the system. It comprises of this sensor array and these counters and all electronic boards and this display. So that's about another type of sensors. Now you see, when this particular sensor is exposed, the initial frequency is roughly, say, 10 megahertz. We use the 10 megahertz crystal, and this trans 2 hexanal vapor is one compound which is responsible for the quality of tea. And when we expose it to when we expose the sensor, which is coated with diphenylaniline, and then when it is exposed, then there is a frequency shift of say around 30 hertz or 30-40 hertz. So if the transreactional is more, then the drift will be more. Okay, so from that we can find out the concentration. So this way we have developed many such sensors. Now about our experience with black tea using electronic nose. And this is the standard technique for testing this tea. And you see we have this is a tea tester. He takes a sip and then declare it not only depends upon the smell or the odor, but it depends upon the taste as well as color. All these factors contribute to the quality. But anyway, we tried the first experiment with electronic nose, and we used the commercially available electronic nose that is from a company called Alpha Moss. And some of these installations are available now in India. Many premier companies like Hindustan Lever or ITC, they are using this electronic nose, but the latest version, obviously. We used it long time back, and we used it at Siri Pilani. There's a research CSIR institute at Pilani. So we took some tea samples to Siri Pilani, and we did the experiment there to check whether this electronic nose is capable of discriminating between the samples. Okay, so we collected the tea samples from the Toklai Tea Research Institute. They supplied this different clones of this tea, so they are of different varieties. And you see, this is the principal component analysis plot. And this principal component analysis, I think most of you may be familiar with. The idea is that, see, we have the 6 sensors in that system. So if there are 6 sensors, so it's not possible to plot a 6-dimensional. Even if you take only one point from one sensor, then it's not possible to visualize the 6-dimensional data. So we reduce the data from 6 dimension to 2 dimension, and then we plot to see whether this electronic nose or the sensor array is capable of discriminating between the samples. So here you see it's possible. So this is the first step. And after that, we have used this artificial neural network. Since we had only few samples, the classification or the accuracy of discrimination we found at 100%. So with this idea, we made the electronic nose of our own, buying some sensors from the market and checking which sensors might be useful for that. So we used 8 such sensors from Figaro, and we made an electronic nose of our own, and we took it to different gardens. These are the names of the gardens, and we checked with the samples, the tea testers, they gave marks. And this is the PCA plot. You see, some of them are coming very close to each other. These are the different marks given by the tea tester. 3 is here, and 4 is also here, 5 is here, and so on. So we could see that roughly it could discriminate, but this principal component analysis is basically a statistical method, and for this type of complex data processing, we need advanced algorithms. So we developed this neural network algorithms, and there are several models we developed, and we found that we could achieve the accuracy to be around 90% or so. So that was our first experiment with electronic nose for the development of tea tasting. But later we found that this tea tasting is really not so easy, and rather we actually focused our research in a different direction. Rather than going directly with the tea tester score, we try to find out some of the concentration of the important markers of tea. So we are now trying to develop different sensors for that. This is another application, aroma profiling during the black tea fermentation. Now, fermentation process is basically an oxidation process during tea processing, and it is during this time of fermentation. I'm sorry. It is during this time of fermentation that these tea samples, they undergo change in color as well as smell. So, and during that time, the grassy smell is converted to floral smell. So there is an important application of electronic nose there, and there are some standard techniques. Colorimetric technique is there, which is standard, which is used by the tea industry people. and we used our electronic nose, and we saw that the results are matching, because this is the fermentation aroma profile, and this is the optimum time of fermentation where the fermentation point should be stopped, so that we could identify very accurately with the help of electronic nose. We did another experiment on fish quality. For the fresh water fish, we used Lavia rohita samples, and we allowed them to decompose for different times, 3.5 hours and 24 hours and 31 hours and so on, and we checked whether our electronic nose could discriminate. But this with this we didn't proceed further because of some other reasons, funding or other reasons. But we could see that this electronic nose could discriminate, so we can assess the quality of the fish, whether how much the freshness of the fish using electronic nose. Now that's about the electronic nose, what we have done. And now I'll give a brief introduction about electronic tongue. And electronic tongue is somewhat better in the sense that in electronic nose, the problem is that since the electronic nose, the number of volatile molecules which are hitting the surface of the electronic nose, that is not constant. So the in any measurement system, the input is there is some uncertainty. Whereas in the case of electronic tongue, since we dip the electrodes on a solution, then there's no uncertainty in input. So that way, electronic tongue is somewhat better, and there are different approaches for designing an electronic tongue, and the terminologies related to tongue is also is well defined. For example, we have 5 basic tastes. They are clearly defined: sour, salty, then sweet, bitter, and umami. So whenever we develop an electrode array, we first check it with the basic human tests, and then we go ahead with our designing of the electronic tongue. So let me quickly go through the details. Electronic tongue, the basic idea is also same. Sorry, the electronic tongue sample. This is the sensor array or the electrode array, and we have the liquid samples, then signal conditioning. It's almost similar to electronic nose. And here, instead of the gas sensors, we use the electrodes. Okay. Now, there are different methods for employing this electronic tongue, and we use the principle of electrochemistry. And there are different electrochemical techniques. For example, the potentiometry is there. So potentiometric electronic tongue is there, where we generate the we measure the voltage when we dip the electrode. But we have used the voltammetric technique, the pulse voltammetry technique, and we have used a potentiostat, and with there's 5 working electrodes. And in this voltammetric method, the voltage is applied between the working electrode and reference electrode, and the current is measured between the working electrode and the counter electrode. So this is basically a 3 electrode system. And I'm not going to details about the electrochemistry. Those who know, it's fine. Otherwise, it's really not very difficult. You can learn on your own. And we apply set of pulses to get the transient response. And from the transient response, we analyze the data and try to estimate the quality. So this is the our electronic node, the schematic diagram. You see, so these are the working electrodes. Okay, so there can be 3, 4, 5, any number of working electrodes, and this is the counter electrode, and this is the reference electrode, and only one electrode is activated at a time, and we apply some pulses, and then we get the transient response with the help of this potentiostat, and this relay box is responsible for activating only one electrode at a time, and we get the data in the computer, and we do the analysis. Data analysis is done, and these are the different types of voltages that are applied. This is called the small amplitude pulse voltammetry. There's a small pulses are applied between the working electrode and reference electrode. This is a large amplitude pulse voltammetry, where large pulses are applied. This is staircase voltammetry and sawtooth voltammetry, and so on. So these are the 5 working electrodes that we used for our electronic nose. These are noble metals. iridium. And the one major advantage of the noble metal is that they are not corroded, and we can wash them easily. We can use it for several times, but they do not have any specificity. That's one major issue. But nevertheless, since we used to get different responses from different electrodes and for different type of voltages, we could get some idea about the analyte on by analyzing the data. Okay, so this is the prototype electronic tongue. All these electronic nodes and electronic tongue have been commercialized by now. This is the GUI. Okay, so and then we have done many other results. I'm just skipping the results with this electronic tongue. Now, in this area in molecular imprinted electrodes, now we are exploring this type of developing of functionalized electrodes. Earlier, we were using since we are basically from engineering background, we use these commercially available sensors and electrodes, and the electrodes are made of noble metal. But we saw that they have limitations. We cannot use them for all for different applications. So we made a team. We made collaborations with different departments. Chemistry people were involved, and we started developing these molecular imprinted electrodes. And molecular imprinting electrodes and gas sensors in this area, it has given a much better success. So this molecular imprinting is basically an imprinting technique. That means we mix a polymer and the target molecule, and after that we make we coat that material on the electrode, and then we wash the target analyte. So there are some grooves formed on the electrode surface, and on those places only that particular target material is absorbed. So that is the advantage of this molecular imprinting technique. Although there are some issues related to selectivity, but we have found that particularly for large molecules, this MIP technique is giving excellent results, because that particular shape that is generated, that is particularly suited to attract those molecules only. Okay, so these MIPs are now being used for several purposes, and we have the expertise in developing this MIP. So this is the idea that we polymerize. This is the target material. We polymerize with a cross-linker, and this is formed. And after forming this, we remove the target material, and after this is formed. So this molecule can only absorb the target molecule. So that is the idea of molecular imprinting. So if you know it, it's fine. Otherwise, you can just go through the basics of molecular imprinting. There are different types of molecular imprinting technique, and we have explored them to develop some sensors. So for example, we have developed these thiaflavin sensors for this black tea. And thiaflavin is a very important biomarker. And you see, this is the structure of this thiaflavin, and we have developed this thiaflavin sensor, molecular imprinting thiaflavin sensor. And this is the procedure that we mix graphite and acrylamide, and this is the target material. This is only 0.05 gram, and this is used as a cross-linker, and then they're vigorously steered, and then polymerization takes place. And after polymerization, we dry it for 2 days, and then we wash it with ethanol water composition, and then for removing this thiaflavin compound, and thus those cavities are formed. And these cavities can only attract, they're expected to attract only these thiaflavin molecules. So this is the basic principle of molecular imprinting, and with that we have developed many such sensors. This is another drawing. I'm just going through it quickly, and this is the shape of the electrode that we make. It's simple. It's just like a pencil. So, and the material is here. So if you dip it, and then apply some voltages, and then with the help of cyclic voltammetry or differential pulse voltammetry, we can get the result, the thiaflavin concentration. So I'm just skipping the results because there are many other applications just to mention. Similarly, we have developed this catechin in green tea using the same technique, MIP-based technique. and just I'm showing the different applications which are possible. And you see most of these molecules are large. We also try to develop some sensors for small molecules, but there the accuracy is somewhat limited. This is epigallocatechin 3 gallate in green tea, using the same technique. All this we have published papers, and some of them have been patented, and we are trying to commercialize now, with the help of some industries. So these are the different methods for making these MIPs. And we have also that's about tea. Let me just go through the next. This is one another application we tried, but we didn't get much success to develop the MIP sensor for pesticides, because the amount of pesticide is really, the range is from the requirement is a zero point 0.1 ppb to 100 ppb. But for that type of sensitivity, it's really very difficult. But there are some papers and there are some methods to develop. We have to work more on this in this direction. So, in another area where we have explored is a quality assessment of mango. Some of the other students, they're working on that. So we have developed the sensors for 3-carine and osimine and furaniol for mango using the same technique. But these are the gas sensor. These are not electrodes. So, and out of these three, this furaniol, the third one, and this is really very interesting. One interesting observation is that we have found that if a mango is artificially ripened, then it is furaniol is vanished. So we have made a furaniol sensor to check whether a mango is artificially ripened or not. Okay, so there are different ways, different applications of this MIP. We can make electrodes, we can make gas sensors. So these are some results and all that. Osimine, this is the osimine sensor, and this is the identification of carbide treated mango through the detection of furaniol. We have done a limited experiment with only a few sample of mangoes, but definitely it has got a good potential. And it's since it's a negative test, we have to explore with many other different types of mangoes and other artificial ripeners also. Okay, so this is all about this MIP. And just another area where we are exploring, and it can also be used for many biological applications. For example, this NIR spectroscopy for agricultural applications. And for this, we have developed one machine. I'm just showing you the machine, what we have done with Toklai. So yeah, this machine we have developed for the assessment of polyphenols in the tea leaves. And this is basically similar to a cement mixing machine, mainly because of the fact that we we have to remove the moisture in tea leaves. So for that, there is a drum here, and that is heated at a temperature of 120 degrees centigrade or so. And these tea leaves are poured here, and then this drum is rotated. And then after the tea leaves are dried, see the drum is rotating, and when the tea leaves are dried, then they're poured on a grinder. And after that, there's a sieve is there, and then after that, there's a quartz plate. And this is the NIR spectrometer, from which we collect the spectra of the tea samples, and we take it to the computer to assess the quality of polyphenol. This instrument is installed in Toklai, and this is quite fairly accurate, almost instant. Only 1 or 2 minutes are needed for assessing the quality of polyphenols in the fields. So there are several other applications of NIR, and for that we use this NIR instruments, the imported NIR instruments from StellarNet of USA. But now, and we have done some experiment on turmeric also, to whether there is any adulterant using NIR spectroscopy. So this is and now we are trying to make the NIR instruments getting the components. This is one NIR instrument. Also, we are trying to develop the Raman spectrometer, because these are low cost, and if you can make it in our lab, it can be field deployable, and low cost definitely. So that's in a nutshell about our research on different agricultural applications, and these electronic nose, electronic and the spectrometers, the MIP electrodes. So now with that, I'd like to thank everyone for your patient hearing. And if you have any question, you may please, Nandita can help me in getting the questions.
Yes, sir. Thank you, sir, for such an informative and enlightening talk. It was so nice. So we have few questions. So I shall put them for you now. So the first question is from Pravat Kumar Swain. He's asking what is the sustainability of electronic nose and its cost effectiveness in present context?
Yeah, actually, it depends. See, this electronic nose nowadays, which are available commercially, they are quite stable, and the sensors, they have a long life because there is a good purging mechanism. See, whenever a sensor is exposed to these volatile molecules, and and there is a possibility that they will stick to that on the surface. So there's a good purging mechanisms are there by which the dry air is flown over the sensors for quite some time, and sometimes heated also the temperature the sensor surface, so that these molecules to go away. So they are used for quite some I mean for several times, and maybe for several years even. This alphamos electronic nose I know, and another electronic nose that is made by our Indian company, that is also giving almost the same result. And cost effectiveness, yeah, that's a very big question, because this electronic nose which are available from these companies like Alphamos or Cyranos, they are very expensive. Okay, and only companies like Hindustan Lever or ITC, they are employing this type of instruments, and the research labs. But the low cost electronic instruments are coming, and because the gas sensors array gas sensors, the price is coming down, and the electronics part is also not very expensive. So miniaturization is now there, and see, every year we see the development in this area. The prices are coming down, the sizes are getting smaller, and there are many other applications of electronic nose. And I hope the generic nose, maybe I don't know whether it's impossible, but even if it is not if it is not there for a particular application, the electronic noses are available and they are now being used. I know this China, they are using this electronic nose very extensively, but in India the awareness or the usage is not that much. Okay.
Okay, sir. So the next question is from Subhash Kanti Roy. He's asking if the tea testing machine is effective for both CTC and leaf tea, and can it distinguish natural and synthetic oil fragments like citronella oil?
Yeah, this is a very good question. See, the electronic nose which are available commercially, they use the conventional sensors. For example, the Figaro sensors, which are not meant for the electronic for the T volatiles, right? But we since we are getting the different patterns for different types of samples, and after that we use some data processing algorithm to find out the this thing, the discrimination. One very interesting story is that when we took our electronic nose in a tea garden in Darjeeling a long time back, maybe 10 years or 15 years back, then one person just for fun put some orange juice. The question that you are asking, the citronella oil, he put some orange juice instead of this tea liquor. So there also the electronic nose gave some reading. So it's really very difficult to make an electronic nose that way with this type of sensors. So for that we need specific sensors. For example, I tell you, this tea research association, they have identified some volatiles for tea. For example, the lilanol is a very important marker, or say geraniol, then transfixin and all that. So if we can develop at least some of the sensors which are very specific to these compounds and make an array of all these sensors, then might be the performance will be better. So functionalized gas sensors or functionalized electrodes are needed, but you have to also see the commercial viability. How many persons will buy that electronic nose? So the person who will investing. So that problem we are facing for this commercialization, it's not so easy. So that's why we are using the generic type of electronic nose, which can be used for tea or coffee or basmati rice, or for any other application. And in that case, this outlier detection, for example, the citronella oil, it really cannot discriminate. It will also show some reading. But if you give only the tea samples, then it will give, correct reading. And for all practical purposes, if you do not have a bad motive, then you should not give this type of inputs to the electronic dose. You will give only the t samples, and you are expected to get results. Okay.
Okay, sir, he is asking another question, like, is your device able to
Next,
detect the
any
quality of
other question?
basmati rice?
Yeah, yeah. We tried to develop one device for basmati rice, and one of the student, did his PhD on this quality of basmati rice. But at the same time, we do not have the sensors. We do not have the Yeah. Next question.
Yeah, so next question is from Shanchari Gorai. She's asking, are the scales for different sensors used in electronic nose same, or which sensor is detecting the smell efficiently?
Yeah, this is a very good question. No, they're definitely not same, because even if you use these MOS sensors, the base resistance is different. Okay, so when a particular sensor is heated to say 300 degrees centigrade, one sensor has got a resistance of 1 megohm, another sensor has got a resistance of 100 kilo ohms. So the their scales are different, and the scales we particularly found it more difficult when we tried to combine electronic nose with electronic tongue. See, for the same sample of tea, we need to get the assessment by electronic nose and tongue, and these scales are they're different. But we have to use some normalization techniques. The data processing techniques are there, by which you can normalize the two different sensors, and then use the yeah. Next one.
Okay, so the next question is from Prabhat Kumar Swai. He's asking, what is the maximum molecular weight of large molecules that can be determined by using MIP technique, or it can be used any macromolecules of the system, or any macromolecules of the system can be used?
Sorry. Uh, um, Dr. Swain, I really do not have the right answer with me, because I'm not a chemist. I have to ask my chemistry sir for this answer. But my idea is that this MIP technique is particularly suitable when the structure of the molecule is complex, or it has got a higher molecular weight. If the cavity formed in that case is truly unique, and it will absorb only that particular molecule. So that's why the performance of this MIP sensor is better if you use it for complex molecules. That's my understanding. But if you want more details, I have to consult my professor of chemistry. Yeah, next.
Okay, so the next question is from Akashlina Marik. She's asking, apart from these wonderful works, you have been extremely active in inventions related to COVID-19 pandemic, one such being the self-powered triboelectric face mask. Could you share your views on that?
Yeah, actually, this is not my idea, but I have become involved with that because this was done by my student. Now he's a colleague, Professor Bipon Tudu. Actually, one day he was thinking that how to, handle this type of, or how to deactivate this type of virus. So he was using this mosquito bats, and from that he got the idea that if you can have a similar type of system. And we had some experience with this triboelectric materials. So one of our colleague, he was working in that direction. So the idea is that if there is some friction between the two triboelectric materials, there is some voltage generated. So if that voltage is useful for deactivating the virus, then that would be great. So that was our idea. Okay, so based on that, we chose some triboelectric materials, and the idea is that there are two layers of triboelectric materials, and when we breathe or when we talk or when we sing, then there's the friction between the two layers, so that will generate some electricity. Now the problem is that since these materials are purely insulators, taking the charge out of it requires sorry, some expertise or some techniques are needed. So we had to develop some using graphite, some conducting paint, and we had to paint these surfaces. And at the same time, we had to check whether there is direct contact or they're getting shorted or not. So all this we have to optimize. And after taking that charge, we put it in between the two wire meshes, and there are small mesh. It's similar to mosquito bat, and they're very close. In between these two, layers, we put one cotton material so that they are not in contact. So we developed after taking the charge, we used one electronic circuit. I'm sorry, I cannot show you the video now. We used an electronic circuit, that's the energy harvesting circuit. So to generate more and more voltage, because we have to give some more voltage so that the virus is deactivated. And then they are connected to the two layers which are conducting, and these are from the thin mesh, wire mesh, made of iron. And the voltage between these two and the gap between these two is a few microns, okay, 100 microns or so. So the electric potential between these two is very high. And when the droplets containing the virus is coming in between, they are getting electrocuted, and they are deactivated. So that is the idea. And we have published the paper in Nano Energy recently, and we have filed a patent, and we are collaborating with one institute in US. So perhaps they will go ahead with the commercialization. Okay.
Okay. So the next question is from Shwetnisha Singh. She is asking, does this electronic detectors are helpful to detect the chemical composition used in fertilizers used for the growth of tea plants or other polyphenols or flavonoids?
No. Actually, we have to use different sensors for that. These sensors are not for these fertilizers. And because for each compound, we have to tune the sensor so that you get more response. So that is the idea. Okay. And that is the research is involved. You have to identify which material is useful for that particular compound. A lot of trial and error is needed for this purpose.
Okay, so one last question is from Shanchari Gorai. She wants to know that if electronic nose and tongue can be related to artificial intelligence.
Yeah, definitely they're related, and we are using these AI techniques for and deciphering the data from this electronic nose and tongue. And unless see, nowadays the idea is that it's similar to our human nose. See, in human nose there are innumerable number of sensors, and these sensors generate a large number of, I mean, data. Mm. And these data are processed in our brain. So similarly, if you can have a large number of sensors, I know in one European Union project, they are trying to develop an electronic nose with more than 1,000 sensors. And see, if 1,000 sensors generate the data, you need some strong data processing techniques to get the information out of it. So advanced data processing techniques, including this artificial intelligence and this deep learning technique and this machine learning techniques, all these are used, and that will definitely make the system robust and much more useful. So I'm very happy that there are so many questions on this, but still we are in the agriculture domain, and we'll be very glad if some of our students. I've become quite old working with this sensors for quite some time, but anyway, I'll definitely be involved with them, and if some idea, if you have, we can share, and you can also develop the sensors for some biological applications. Okay.
Yes. Thank you, sir. Thank you so much for taking all the question, and thank you so much for sharing your research work in our platform. It was such an informative talk, and thank you again for joining us, everyone. Join us again on 24th Jan 2021 for our regular lecture. Please follow us on Facebook and Twitter, and please subscribe to our YouTube channel, and do check our website as well. Our previous talks are archived in our YouTube channel and website, so you can check them out there. Thank you again for joining us. Take care and stay safe.
Okay,
Thank
thank
you,
you,
sir.
Nandita, for organizing this seminar and moderating in a nice way. Mm.
Thank
Okay,
you, sir.
okay then. Bye bye. Mm.
Thank you, sir. Thank you. Thank you so
Mhm.
much.
Okay.